And then let me stop the recording.
Shall I leave mine or take it?
Just leave it there.
So
Okay,
it says
good
that
morning,
the
everyone.
meeting is recorded. It's being recorded.
Yes, is that okay?
What can I do?
Okay, good morning, everyone. It's our first Monday in April. It's feeling in the Northern Hemisphere, a bit like spring. And so with all its rejuvenation, and we hope rejuvenation of the planet at the same time. Anyway, we have two great speakers today. And without further ado, I, Bob Sinclair, will hand the baton over to Professor Wa Chu, who will introduce our first speaker from Israel. Wa, please.
Okay, good morning, everyone, and good afternoon and evening, wherever you are. It's my great pleasure today to introduce my friend, Professor Ada Yonath from the Weizmann Institute of Science in Israel. I have the fortune to work with Ada some years ago when she began the work on ribosome structure analysis for both Cryo-Em and X-ray crystallography. And I remember vividly the enjoy experience that I had when I worked with Ada, both in Europe and as in States. Her full engagement, compassion, and determining the crystal structures of ribosome was unbelievable and incredible. And finally, she was able to succeed. And in 2009, and she shared the Nobel Prize with Tom Stiles at Yale and Venki Ramakrishna at the RMB in Cambridge of the Nobel Prize in chemistry. And we were very excited all this year. I always admire Ada's enthusiasm and the insightful aspects of macromolecule structures. And today she's going to give us a lecture on bridging between prebiotic life and acute problems in modern medicine. I look forward to hearing your lecture, Ada. Thanks very much for coming.
So do you hear me?
Yes.
Yes.
Do you hear me?
Yes, I hear you.
Thank you for inviting me. Thank you for inviting me. It was always a pleasure to interact with you, Wa Chu, and with Stanford, and at the synchrotron of Stanford at SSRL. I enjoyed it all. And today I will tell you about bridging between prebiotic life and acute problems in modern medicine. Actually, I'm also talking about bridging between X-ray crystallography and Cryo-Em. So you all know that the DNA has the genes. Each gene is coding for a protein. DNA has much more, but it doesn't matter right now. The information about the protein sequence is in the DNA, and it's not useful because it is not exposed, not readable. So first of all, it's being transcribed to messenger RNA, which is a very similar molecule. I think you all know that. And then it's being translated to proteins by the ribosome. So there is a huge number of ribosomes that function in each living cell. Mammalian cells may contain millions of ribosomes, like in the liver, 5 to 6 millions. Even bacterial cells contain up to 100,000 ribosomes when the bacteria divides. All ribosomes perform their two major functions. It means the decoding and the peptide bond formation. It means the creation of the protein. In the same way, it doesn't matter if it's bacteria or elephant, they work the same. The ribosomes have evolved, but not the active sites. In vivo, the ribosomes act continuously, and they can make up to 40 peptide bonds in a second. Hardly make mistake. Mistake rate is about 1 to 1 million. So actually what happens, the ribosome is made of two subunits, a small one and a large one. The tRNA that brings the amino acids binds to the small one, to the messenger RNA, binds or at least interacts, and the peptide bond is being made at the large subunit. The interactions between them are also through their own body, but tRNA is a major component. Actually, there are 3 tRNA sites. So if we look at the ribosome from top, this is the small and the large subunit. Messenger comes in, the tRNA brings the amino acid from here, and there are 3 sites on the ribosome, A, P, and E. A, the amino acylated tRNA is interacting here. P is where the decoding happens, and the peptide bond between the A and the P. The protein goes out through a tunnel when it grows. And after the peptide bond is being made, the P site tRNA became empty and goes out to the E site, exit site. So A, P, peptide bond formation, PTC, it's called the site, and the empty tRNA leaves. That's all. So in the beginning, we worked only on bacterial ribosomes, normal bacteria, and we know now it's nothing, but at that time it was a big thing, as you've just heard from Arju. We know the position of each and every atom. This we determined at 2000, 2001. You see here the small subunit on the left and the large on the right. They are made of RNA, that's the gray. Most of the ribosomes are made of RNA, except that in the mitochondria, and many proteins. So the proteins, each of them is shown in another color. So actually what we talked about until now, small subunit, large subunit, PTC is where the peptide bond is being made, and the 3 sites are here. It means these are the 2 active sites of the ribosome, main active sites, and the tRNAs are shown here. And that's a view from into the active sites, active regions. The peptide bond formation is a very simple reaction. A site tRNA comes with an amino acid on it. I hope you can see. And P site is the extending protein. And this happens here. The bond is between the NH2 of the incoming amino acids and the growing protein. And the tRNA, just to remind you, is always an L-shaped molecule, very highly conserved. The piece that is always the same, fully conserved, is the lower part, which carries the amino acid. So it always CCA amino acid in every cell, in every animal, in every bacteria, whatever. And the decoding is always here, free nucleotides that point out of the double helix. So what we learned, looking at the structure, is that the ribosome is actually not doing any active catalytic event. It just provides the stereochemistry that is needed to make the peptide bond. So here is the A site tRNA and the P site tRNA within the ribosome, which is a gray, and I will keep the same colors for the next few slides. The tunnel is here and peptide bond is being made here just by a frame, the frame that allows it. Peptide bond formation is an exothermic reaction, so it can take care of itself. This is, when we looked here, we looked at the tRNAs, but also when we look at the products or at the reagents, you can see that they also sit in a way that they can make the peptide bond. So it doesn't matter if it's a very long component or short one, or a substrate that grows out, doesn't matter. The piece that is that has to make the peptide bond or is in the position to make peptide bond is sitting in the sort of semi-symmetric pocket by the ribosome. Sorry. So for this structure, for this part of our studies, we needed many structures, as you could see, many structures, and we used also structures from other universities like those from Yale, from Stites group. But anyway, we still needed many structures. So it took 20 years for the first structure, 21 years for the second, 3 to 5 years for the following. And now with cryo-electron microscopy, we can obtain structures within weeks or months. So owing to the specific properties, our current studies are hybrids of both of crystallography and cryo-temperature, cryo-Em, cryo-electron microscopy, and I will explain it later. But let's go back to the pocket that makes the peptide bond, or the peptide bond is being made It's made of two semi-symmetric pieces that you could see until now. And as I said, I will keep the A site in blue and the P site in green. And this is the way it looks in the contemporary ribosome. But actually it looks like this in all ribosomes, all known ribosomes. And the sort of symmetry, semi-symmetry is like sort of unseen axis here. So if we look at this pocket from top, and what I did here, I put one on top of the other of the active site from eubacteria, archaea, eukaryotes, mitochondria. You can see here the PDB numbers. After I put the main chain, I couldn't distinguish between them, and each of them has another color. So this is very highly conserved pocket, almost fully conserved, even in terms of sequence, and I'll come back. But I come back to my main I want to remind you my main slide. We needed many structures for coming up with this idea. The sequence, as I said, is conserved almost fully. If we look at all the nucleotides, they are at least 50% conserved, but mainly between 95 to 100%. And even when they are not fully conserved, purines are replaced by purines and pyrimidines by pyrimidines. So the structure is kept. So the high conservation of the symmetrical region indicates that its existence is beyond environmental conditions. And therefore we propose it to be in the proto-ribosome. So what do we mean by the proto-ribosome? A prebiotic bonding entity termed by us proto-ribosome, which is still functioning in the contemporary ribosome. So we go now back to prebiotic time. So we assume that the ribosome originated within an environment which could contain elements of life, and that the prebiotic chemistry could generate and elongate RNA chains. So we decided to make lab constructions of the proto-ribosomes. If it could be a machine in the past, we decided to use the knowledge from the contemporary ribosome to do it. This took years, almost a decade. But now we can make a lot of constructs that resemble the pocket. And when we made it, our hypothesis was that the catalytic proto-ribosome is a product of dimerization, and it requires the existence of self-replicating, self-folding, and self-dimerizing RNA molecules. So the first one, and afterwards now we have many more, the smallest one is two chains of 67 nucleotides, but we have also some that are about 90 nucleotides. We could characterize the production of the D peptide by MALDI, and we had controls, negative and positive, you can read here, and the time is less than a second. So to sum up, our hypothesis is based on the existence of self-replicating RNA molecules with catalytic capabilities, and that's now very important, on the assumption that the genetic code followed its products. And I don't think that now I can discuss it in detail. So again, coming out of it, based on the suggested existence of RNA-dominated world and our experimental results, we propose that the proto-ribosome is not only the beginning of the ribosome, but it's the entity around which life has evolved, life in total. And again, as I said earlier, we needed many structures, and in the beginning it took years, now we do it in months. So let's look at the ribosome for a minute and compare. For instance, motions of the ribosomes. When the E site tRNA exits, now we know that there is motion. We actually knew it earlier because we had the structure with and without exiting tRNA, but both were crystals, and neither of them were very clear here in this area where the E site tRNA goes out. So what you see here is a hybrid of crystallography and Cryo-Em in the two colors of the same large subunit of Staph aureus, which is a pathogen, at similar, not exact resolution. And you can see that only in the EM we could see this part, like it is hardly connected. The rest is very similar, and this is where the E site lives. So we think here there is a very important helix, RNA helix, H68 the name is, and this is the stalk. And we think that there is, we thought, looking at these maps, that there is a motion here. So we collected data of several, I think nine substructures, statuses, in between situations. To remind you, we look at it here, and we could see the motion in the EM, Cryo-Em structures. You can see here, and even this one. So this, at the end, this goes here. We could follow the exiting tRNA in detail. So let's talk a few minutes about antibiotics. The natural antibiotics are the weapons that one microorganism from one type are using for interfering with cell life of different species. And owing to the vital role played by the ribosome, over 40% of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. So I will not show this now, they bind to the active sites. Like you see here, the large on the left and the small on the right. And all these spheres are showing a family of antibiotics. So what do I call family? It's the natural antibiotics, and what the company is made to make them better, faster, less poisonous, whatever. But they bind to these positions, which are all belonging to that active sites, like the decoding, the hinging, these motions, the bonding, and to the tunnel. So let's have a look for a minute, a little bit more detail. Those that are called macrolides are coming out of erythromycin, and they bind to the tunnel. And you can see here two types, two different macrolides, and see how nicely they block fully or partially the tunnel. But even when they they block partially, protein will not be able to go from top to bottom. And one of the most important nucleotides from the ribosome that interact with them is this one, which is A in position 2058 in E. coli, and becomes an N in human with evolution. So this is why these antibiotics don't bind to human or to eukaryotes in general. So I don't want to go into detail. I just want to show you how it sits, in great details. How could we see it at the different crystal structures? And now we can see it also by cryo-electron microscopy. But I want to talk more about resistance, which is one of the most severe problems in modern medicine. So the clinical usage is disturbed by resistance because the usefulness is depending on small changes, as I showed you a minute ago. Also, not all the resistance mechanisms, but many of them are based on small changes, like the bacteria acquired the knowledge. Yet, there are some resistances that are depending on remote changes. And this, for me, was a fantastic discovery. So what you see here is the small subunit and the large subunit and the free tRNAs. The purple one is just living, and protein is made here, will go through the tunnel. And this is the position of erythromycin, which I mentioned earlier. The tunnel is made mainly of RNA, but there are free proteins that bulge into the walls of the tunnel. And we found that one of the mechanisms that are acquiring resistance is not happening here, as I showed you earlier, at the position, but far away here at the end of this protein. So actually we are looking a little bit more in more detail. We are looking here on the large piece. Do you see my, oh, okay, it came. And this protein doesn't even touch the position. Yet, changes at the end of the periphery of the ribosome made changes in the loop that comes up. I cannot go now into all details, and I'm not sure that you really want to know all of them, but these are the two loops, the coral that shows the normal, the apo, and the blue or gray that shows what happens, and you can see how it moves from the end of it, and until it comes to the tunnel, it makes this little change that in the erythromycin binding place, and this is what acquires resistance. So resistance can happen by mutations really far away, over 25 angstroms away. So again, for this, we needed lots of structure, and we are using a hybrid. So the increasing appearance of multi-resistant strains together with the minimal, actually negligible number of new antibiotics by pharma companies brought the World Health Association to say that looking at the numbers, 2 million people fall ill every year, or in Europe, 33,000 deaths because of resistance, although they invested 1.6 billion healthcare euros and productivity loss and all those brought the World Health Organization to say that we are reaching an era that people will die from pneumonia or wounds in no time. And even the World Bank estimated that almost 4% of the global economy will be lost by 2050 because of resistance. Yet, as I said earlier, hardly any activity by companies in looking for new antibiotics or designing new antibiotics, because of the big mismatch between the investment and the expected profit. So on the left here, you can see how from 1990 to 2002, 12 years, what is the growth of resistance of 3 different bacteria, 3 pathogens. And on the right side, you can see the number of new antibiotics from 1980 to 2014. It means much longer time and hardly any new antibiotic. So a question that we asked ourselves, can we combat resistance in full? So in our opinion, it's unlikely because bacteria want to live and because bacteria are cleverer than us, at least in terms of survival. Nevertheless, we decided to look into it with the concept that we may be able to identify pathogen-specific essential structural motifs that do not belong to the ribosomal active sites, and therefore maybe they can become sites for new antibiotics. So our modest contribution is by determining the structures of ribosomes from resistant pathogens. And we did it, again, we needed many. So I repeat the same, we did it in a hybrid way. So we determined the structures of ribosomes from pathogenic bacteria and compare them to harmless, the ribosomes of harmless. So the generalization is that all bacterial ribosomes are similar, but not identical. And we have many lessons. I'm not going to talk about all of them, but lesson number 4 is beyond my expectation. It highlighted novel antibiotics binding sites. So if we look here, we see the skeleton of the large subunit in gray, and what is addition in Staphylococcus in cyan. And I show here only those that are on the periphery. And you can see they are on the periphery, they have their own structure, and it is different than the same place in the, what we call normal ribosomes. So if we focus on one of them, let's say on this one, we can see that it's in the environment is very similar, but this part is bulging out and it has its own structure. So it may be a potential binding site. Actually, there are many potential binding sites. You can see here all these. In a model system, we identified 25 new potential unique sites. We block them one by one, and 16 of them cause a stop of cell growth. And because these sites are not involved in primary ribosomal activity, there is no genes against them. So for now, these sites can be exploited for the design of advanced degradable antibiotics. Why degradable? Because the antibiotics that are now used are not degradable. I'll come back to it in a second. But first, I want to say that the new antibiotic may be different, may differ or may distinguish the pathogens from the microbiome, which are the good bacteria in the body, and these good bacteria can easily be hurt by the normally used antibiotics. So it is a problem right now. But the new ones, because we designed them, we can make them that they won't look at the microbiome. They will be also distinguishing between the host and the pathogen, because most of the difference between new bacteria and eukaryotes are on the surface. You can see here in red or in blue and green. I don't want to go into detail, but these are the differences. So we can do this, and we can also look, as I said earlier, as I promised you, environmental and ecological conditions. So most of the ribosomal antibiotics are extensions of small. They have in the core small molecules that cannot be digested and they cannot be degradable. And therefore, when we take antibiotics, us or animals, even in small amounts, these species are coming out, and because they are not digestible, but because they are not degradable and too small for most of the purification systems to see them, they can sneak into the agricultural irrigation system and come back to us, increase resistance, and may do many other bad things. So I just say that we are looking now for pieces on the surface. I showed you one side, but actually, if you want to look at details, there are here some. I don't want to discuss each, but each of these are specific to staph aureus. So these are target sites that we are working on now, but also we are working on tuberculosis. And tuberculosis, if we compare tuberculosis to other ribosomes, the large subunit has this huge extension, which is specific, and we can try to target this one, like you can see here. So of course we couldn't do all these studies if we had to crystallize and work the structure without EM. So I think I can just say this, I can summarize this, because we are designing the newly designed, the next generation antibiotics. We can make them so they are, we can handle them. So far we were using either nucleic acids or proteins or oligopeptides. And this can be optimized, so we can be in charge as much as possible in their chemistry and their fate. So in summary, the new insights obtained from these high-resolution structures of the ribosome from genuine pathogen provide unique chemical tools. I didn't show you several, but I showed you for suggesting novel sites. They also suggest how to improve and how to get better. I don't want to discuss all, but the most important thing is the design of pathogen-specific antibiotics. So we are aware that the companies don't want to do it, but maybe with time they will change their mind. So there is another point that we looked at is ribosomes hibernation. So owing to the vital role played by the ribosome, earlier we talked about antibiotics, but also cells developed a specific method for their preservation under stress while not operating. So if there is stress and the ribosomes don't operate, actually they would be digested and gone. And if the stress is long, like the whole winter or something, the animal can, or the cell can get up without ribosomes, it means death. So the cells devised a method to make dimers. You can see here one ribosome and another ribosome. This is in bacteria, and they block each other. They block the entrance of the messenger RNA. So they keep each other intact, but not active. So we have seen this structure not only in one bacteria, but in 3 of them, Staph aureus and E. coli, and we put them one on top of the other, and you see that the dimerization is different, but it's the same one. And of course, we can think about using this for antibiotics also. So a few words about Cryo-Em. You know that in 2017, the Nobel Prize in Chemistry was awarded to Debouchet, Joachim Frank, and Richard Anderson. And the explanation, I like the explanation of the Nobel Company, or the Nobel Committee, for developing cryo-electron microscopy for the high-resolution structure determination of biomoleculars in solution, not in crystal. This is the biggest contribution of Cryo-Em, of course, time and so on. We don't need to grow crystals, but we have to collect a lot of data. So here you can see the procedure in glance. We can look at many, actually here, we can look at many positions of the molecule in the experiment that may have many different conformations, especially the ribosomes that is very mobile. And then we can group them and we can look at the groups, and by a very ingenious treatment, we can get at the end a model, and sometimes a structure, sometimes several structures, if the population is very large and if the molecule is very active, very dynamic, and we can see them. And in all, we can, maybe you cannot see it here, so I will show it in a minute. In all, we can decide what is the actual resolution of a part, of a subpart of the molecule. So for instance, ribosomes, you can see here a chart that shows that although the best resolution here was 2.2 angstrom, almost the atomic resolution, there are regions that have 5 angstroms, and these are mainly on the surface. So that's a I don't want to call a problem, but this is the character of the Cryo-Em results. Recently, this year, came out 2 papers that talk about Cryo-Em atomic resolution, one by Holger Stark's group, and you can see here how wonderful the maps look, and the other by Sherris, that a much larger group. See, the whole slide is their names, but it's not important. What's important is their maps, which are, in my opinion, mind-breaking. So they can see even water and hydrogen bonds and so on. But even here, even in a structure that came out from Sherris group that started for many, there are several substructures, and they made a specific comment that the surface is much less well-defined than the rest of the molecule. Here again, you have the chart. So this is a characteristic of Cryo-Em that one can deal with it, but one has to remember it. Actually, structure determination at cryo temperature started in Stanford. This is me, you can see here, not very hopeful. And this is the experiment where we did first time X-ray crystallography at cryo temperatures in Stanford at SSRL in 1986. Although people thought that it won't work, and even I was not very hopeful, it worked. And one can see from the number of PDB depositions, here is the cryo temperature was introduced in the red, where the red is shown, beginning of 87, we did it in November. And until 2008, what I show here, we had almost 50,000 structures, whereas before this, they were less than 100. And it grows, it grew. But let's see what happens after 2008. So here you can see the number of newly determined structures. 2008 is somewhere here. Oops. And today, the number is so large that 2008 looks like nothing. And that's not all by crystallography. In the lower part, you can see the amount of structures that came from EM in yellow. Crystallography is still in blue. The numbers in total, they grew, but the electron microscopy contributed a lot and now it's clear why. So before I finish, I want to thank the Weizmann Institute that let me work on this crazy project. The president that made the decision was biologist, Michael Saylor. He still is a biologist. And people say that because of it, he tolerated me. But when his time was over and he was replaced by Chaim Harari, a high energy physicist, he didn't kick me out. And this is because the Scientific Advisory Committee, Kendra and Vincent and Rich, say that a project like this should not be stopped, although it's not clear that Ada will make it. It all started with a very strong collaboration with Max Planck, with Weizmann in Max Planck in Berlin, that his dream was to see the ribosome in function. He died 10 years before the ribosome came, and he was replaced by Franceschi and then by Fucini. But what's important is that he established a research group for us at the synchrotron in Hamburg. Now the synchrotron in Hamburg, Daisy, is giving synchrotron beam, but also Cryo-Em. They have a fantastic arrangement for visitors that want to use the Cryo-Em. I'm not there anymore, but I admire this. So I want to thank my two groups, my two research groups, for their devotion and determination in good and in bad time for many years. I want to show you the group as it is, actually not as it is now, because now only the green stars are, but this is the group, and this is where we collected data. The group is run by Dr. Anat Bashan. She is the real scientist that runs it. I'm just talking to you while they are working. And please pay attention to Tamar. She came 19 years ago for 10 weeks. She's still around, and she baked a cake. I took a picture in her birthday, so she baked a cake that looks like that. It shows that in my group, the ribosomes are considered sweet. I want to thank my family. I cannot show my sister and my mother and others, but you can see here my daughter. She's a chief MD, and her daughter, that at the age of, she was at that time 9, she made a speech about me, and she said, as you know, she is a very busy scientist, but she always has time for me, and she gave me a prize. That's not her prize, that's the Nobel. For me, her prize is at least as important. The grandma of the year is Ada Yonath. She didn't write which year, so I asked her which year, what's the prize, every prize is a year. And she said, you have to prove yourself every year, otherwise I take it off the wall, and until now it's on the wall. It's already 13 years. Ribosomes became popular. That's a carnival. He is me and his wife. It's a ribosome, you see. Protein comes out of him. And Kishka, Michel Kishka, one of the most admired artists and writers in Israel, drew me. He's not a crystallographer. He's not a scientist. He found it he said he found the structures in Google. Small subunit, large subunit, small, large, small, small, fully symmetric. And he thinks that the new antibiotics should be here and here. And thank you very much.
Ada, thank you very much for these phenomenal lectures that bring from the prebiotic life to the current life. It is incredible. And from crystallography to Cryo-Em, this is remarkable journey that you have shared with us today. And I think in the interest of time, I would suggest if you can stay online until the next lectures, and with all the questions posed by the audience that you can answer. Okay, let's give a big hands to Ada for this really, really exciting talk. Thank you very much, Ada. Okay, good. So we will.
I will try to get out. Yeah.
Yeah. Good.
Stop share.
Yeah. Stop share.
You don't need.
Yeah, stop share, right?
You don't need it anymore, correct?
That's correct. Yeah, that's great. Thank you. Okay, back to Bob.
Okay, thank you, Ada and wow, fabulous. Somewhat makes me apprehensive about all these bacteria, but maybe we can discuss this later. There is many questions in the Q&A, and we will transmit them to you if we cannot answer them in the post session after the next talk. Anyway, I'll pass on to Professor Yiche, who will introduce our next speaker, please.
Hi, good morning and good evening, everybody. This is Yiche in material science here at Stanford. It's my great pleasure to introduce Rafal Dunin-Borkowski. He's currently the director of Institute for Microstructure Research and the Ernest Raska Center for Microscopy and Spectroscopy with Electrons. Well, he's very well known. You know, I don't need to go into detailed introduction. Let me just mention the few awards he won. He was awarded the Raska Prize of the German Society for Electron Microscopy, and he received the Advance in Synergy grants from the European Research Council. We know Rafal has been a leader in high resolution electron microscopy, particularly go deeper into the, starting spin, starting the, the quantum physics of that. And he has a lot to say, to contribute. He's building up this big center in Germany. With that, let me pass this to Rafal.
Thank you very much, Yi. I hope you can hear me.
Yeah.
Great. So first of all, it's a great honor to be speaking to you today. And now we switch subjects to in situ transmission electron microscopy of functional properties and dynamic processes in materials. So this is mainly a talk which focuses on material science and physics, but I will also touch on some topics related to electron beam sensitive and maybe biological materials. The picture on the first slide shows some images of magnetic fields that we've recorded, which we don't yet fully understand, but I'll come to them somewhere halfway through the talk today. So as you mentioned, I work in the Jülich Center in Germany. And on the left is a map of Germany. The blue squares are different research centers of the Helmholtz Association, which operates large-scale devices and infrastructure. So Jülich is marked with the red circle. It's about 3 hours from Paris and about 2 hours from Frankfurt. And I'll explain later why we refer to electron microscopy as a large infrastructure in Germany at the moment. So from above, Jülich as a research center is located in a forest, and our building, the Ernst Ruska Center, where the electron microscopes are housed, is marked again by the red circle. It looks quite idyllic, but this image also highlights the importance of multi-scale characterization, because if you change the field of view and you still see where the Ernst Ruska Center is located, it's in fact located in between two of the largest coal mines in Europe, just a few kilometers away. And these coal mines operate 24 hours a day with large bucket excavators. And so this has implications for the stability of our electron microscopes and for the size of the anti-vibration systems that we have to install underneath them. So this is the building where we house our main electron microscopes. The two towers house the two largest instruments that we operate. We currently have 7 aberration-corrected instruments. We have a strong focus on methodology and instrumentation development, dating back to the development of spherical aberration correction for transmission electron microscopy by my predecessor Knut Orban, together with Harold Rosa and Max Heider. We've been operating as a national user facility for 16 years, and we have a spherical and chromatic aberration corrected microscope that we call the Pico Microscope. And this is very similar to the team microscope in Berkeley, with a slightly different version of the chromatic aberration corrector. And this slide shows some people you may recognize standing in front of this microscope, which is 4.5 meters tall and higher than a normal high base Titan microscope from Thermo Fisher. So today I wanted to concentrate on 4 topics. First of all, I wanted to describe some work on characterizing magnetic fields in materials, both in projection and in 3 dimensions. This will be the main part of the talk. Then I wanted to describe some recent work on switching of very small nanoelectronic devices in situ in the TEM. Then I will go on to show a few slides on imaging electron beam sensitive materials. And finally, I'll describe a few perspectives and plans for the research that we're doing in the Ernst Ruska Center in Jülich. As I mentioned at the beginning, the main focus is on dynamic processes and functional properties. And these will be studied using both phase contrast and diffraction contrast techniques, and both TEM and STEM techniques. So first of all, I'll start with magnetic fields. And magnetism is, of course, a very important topic across many different scientific disciplines, ranging from biomagnetism to rock magnetism to magnetic storage, to magnetic molecules and magnetic catalysts in chemistry and so on. And in most of these disciplines, we are either interested in characterizing magnetic fields represented by the magnetic flux density B, or by the source of the magnetic field represented by the magnetization M. And of course, we'd like to characterize magnetic fields on the nanoscale, either in projection or ideally in 3 dimensions, and ideally with close to atomic spatial resolution. This is extremely challenging using any technique, and in particular, most magnetometry techniques, which are carried out on bulk samples, provide relatively little information about the nanoscale magnetic microstructure of the materials. So magnetic imaging in the transmission or electron microscope can be carried out using a few different techniques. Some of these are spectroscopic techniques. In particular, in eels, there is electron magnetic circular dichroism, which is sensitive to the magnetization M. Normally, the magnetization measured in the direction of the incident electron beam. And then there are phase contrast techniques. And these techniques are usually sensitive to the magnetic flux density B, and especially the magnetic flux density oriented in a direction perpendicular to the incident electron beam direction. And here we have some TEM techniques, for example, the Fresnel and the Foucault modes of Lorentz imaging. This is effectively defocused imaging and dark field imaging of magnetic microstructure. We have off-axis electron holography. And then we have STEM techniques, which can be used for imaging magnetic fields, especially differential phase contrast imaging pioneered by people like John Chapman and Joe Zweck. And then more recently, there are 4D STEM techniques, including STEM tychography. None of these techniques is better than any of the other ones. They are each applicable to different types of sample and different types of imaging. The one point I would make is that most phase contrast techniques are applied with the electron microscope objective lens switched off, so that the sample is in magnetic field free conditions, and instead usually a non-immersion lens is used instead, and then magnetic fields can be applied to the sample to study switching processes in situ in the electron microscope. In Jülich, we focus on off-axis electron holography, primarily because this is one of the best techniques for imaging magnetic fields in very small structures, especially small grains below about 100 nanometers in size. And here we use a low base Titan electron microscope, for which the ray diagram for off-axis holography is shown on the left. And here I'd like to thank Molly McCartney, who taught me electron holography when I worked in Arizona State University in 1997 and 98. The ray diagram for off-axis holography is very similar to that for bright field imaging, except that some of the electrons that go through the sample are bent towards electrons that pass outside the sample with one or more electron biprisms. These biprisms are just very narrow metal or metal-coated quartz wires that have a voltage applied to them, and they introduce a triangular phase shift to the electron wave function. If the electron source is coherent enough, especially if it's provided by a field emission source, then where the electrons that pass through the sample are overlapped with electrons that passed outside the sample, there is an interference pattern formed, superimposed on a normal bright field image of the sample. And this interference pattern is the electron hologram, which carries direct information about the phase of the electron wave. In other words, it solves the phase problem in electron microscopy, and it provides access to all spatial frequencies of the phase, including low spatial frequencies of the phase faithfully, in addition to providing a perfect energy filter in the electron microscope. For imaging magnetic materials, as I mentioned, the sample is located in magnetic field-free conditions, and then an external magnetic field is usually applied using the vertical field of the objective lens, and then the sample can be tilted in that field. So the phase shift is important because it provides access to the projected electrostatic potential or the in-plane magnetic field B projected in the electron beam direction. And today we're focusing on the magnetic field. Just as a brief older example, this image shows a typical off-axis electron hologram of a multi-walled carbon nanotube. The bright band of contrast is the overlap region of the wave that passed through the sample with the electron wave that passed outside the sample. And by taking a hologram, recording the magnetic contribution to the phase shift, and then displaying the cosine of a certain multiple of the magnetic contribution to the phase, the magnetic field inside and outside the sample is displayed with nanometer spatial resolution. In this case, again, these are the in-plane components of the magnetic field projected in the electron beam direction. So one of the challenges with imaging magnetic fields is that we have a three-dimensional magnetic vector field, which is being projected onto a two-dimensional plane. And this is one of the challenges with interpreting off-axis electron holograms of magnetic materials. So just to begin with, I'd like to illustrate why we want to image magnetic microstructure in a real material. And this is a magnetic high entropy alloy. Now, high entropy alloys are metallic alloys which are formed from near equiatomic proportions of 5 or more elements, and they have tunable magnetic properties depending on the constituents of the alloy and also on the preparation conditions, especially the annealing conditions. And in this slide, what I'm beginning by showing is a hysteresis loop, which shows magnetization on the vertical axis, applied magnetic field on the horizontal axis for two different magnetic alloys. A soft magnetic alloy of aluminium, cobalt, iron, nickel, which is not a high entropy alloy, shown in red. And then in black, this is a hysteresis loop for a high entropy alloy, which is the same material, but with some of the cobalt substituted for chromium. What we'd like to understand is why the high entropy alloy, which has chromium in it, has a lower saturation magnetization and a higher coercivity than the aluminium, cobalt, iron, nickel alloy. So the reason it has very different properties becomes apparent if we first image the physical microstructure of this alloy. And in this slide, I'm showing a high-angle annular dark field image on the top left. And this high-angle annular dark field image reveals that the microstructure is incredibly complicated. Surrounding this high-angle annular dark field image are chemical maps recorded with EDX of the different constituent elements. And they show that the microstructure has many different levels of hierarchy. It has small precipitates, it has larger regions of different composition. And if you look at the pink map, which is the ion distribution, this is the map. This is a map which shows the regions of the material that are more strongly magnetic. If you look at the region which is nickel rich in the cyan color, it shows the regions that are more weakly magnetic. It turns out this microstructure contains two different characteristic regions: a weakly magnetic region with strongly magnetic precipitates of two different sizes inside it. This is the region which is labeled R1 on the right. And it also contains regions which are strongly magnetic with very weakly magnetic void-like regions inside it. This is the region which is labeled R2. And the reason that the magnetic properties are so complicated becomes apparent when we image the magnetic microstructure with off-axis electron holography, because whereas the R2 region, which contains the strongly magnetic phase with the weakly magnetic void-like regions inside it, shows large magnetic domains that are relatively simple. In the region outside it, the region which is labeled R1, which contains lots of little precipitates, in fact, the holography reveals that every one of these little precipitates contains a magnetic vortex where the magnetization spins either clockwise or counterclockwise. And when these precipitates are imaged in the presence of an applied magnetic field, then every one of the precipitates reverses magnetically at a different value of the externally applied field. This is information which would be almost impossible to record using any technique outside the electron microscope, and off-axis holography is one of the unique techniques for recording this information inside the microscope. And so what off-axis holography tells us for this material is shown schematically on this slide. On the left, we have color-coded in different regions of gray, regions which have different saturation magnetic inductions, which are ferromagnetic to different extents. So there are at least 3, perhaps 4 regions which have different magnetizations inside the material. They are subdivided into precipitates of different size. And on the right-hand side, what off-axis electron holography tells us is that these different regions have very different magnetic properties. Some have large magnetic domains and some have vortex-like magnetic states. And the reason the coercivity is so difficult to understand results from the complexity of the magnetization reversal process through this physical and magnetic microstructure, which involves successive reversals of the different magnetic vortices in the different precipitates. The other magnetic system which I wanted to describe is a system which we've been looking at for 4 or 5 years now, and this is magnetic scomions. These are magnetic structures which are of interest for magnetic storage and magnetic logic applications. These are vortex-like magnetic states which form typically in crystallographically chiral materials, both in bulk-like materials and in thin films, and they take 2 very characteristic forms, which are referred to as block-type and nail-type scomions. In the presence of an applied field, usually at low temperature, most of the sample is normally oriented in the direction of the applied field, in the direction of the small red arrows in these diagrams. But then in the centers of these little vortex-like structures, the magnetization points in the opposite direction. In block-type scomions, in between the magnetic moments fall into the plane azimuthally, whereas in nail-type scomions, the moments fall into the plane radially. And I'll concentrate on the left-hand case here, the block-type scomions, in the next few slides. So I'll start with an example which shows some work that we did a few years ago in 2017, where we patterned a crystallographically chiral material iron germanium into a long stripe. And this is representative of a magnetic track in which we may want to nucleate magnetic scomions, and we'd like to see how they travel along this track. So this track is 45 nanometers high on the left, 150 nanometers high on the right, and it's about 100 nanometers thick in the electron beam direction. And in this case, I'm starting with a bright field TEM image, but what I'll show next is magnetic induction maps recorded using off-axis holography at low temperature in different applied fields, and the field is applied out of the plane of the sample in the electron beam direction. So now I show the results of magnetic states recorded in the region of the magnetic stripe shown with the white dashed rectangle in the bright field image at the top. In zero applied field, we have what is called a helical magnetic structure, which extends right to the very edges of the magnetic track. And then in the presence of an applied field, a single row of scomions forms. These are vortex-like structures which all spin in the same sense. But if you look at the edges of the stripe, they are either red or green, and this is referred to as a magnetic edge twist. It's a signature of the fact that these are magnetic scomions. The scomions are squashed vertically on the left, and they're elongated vertically on the right. And then in the presence of a stronger applied field, in the image at the bottom, the scomions start to form a zigzag array. And all of this can be imaged in real space with nanometer spatial resolution. The reason I show this is that these are really three-dimensional magnetic states, and we would ideally like to be able to image the three-dimensional magnetic structures directly. And I also wanted to show a few other examples which illustrate why we want to image three-dimensional magnetic states. And the examples which I have on the next few slides show that in some cases we have a mixed block type and nail type character which we'd like to image in 3 dimensions. In some cases there are magnetic singularities. In some cases there are magnetic superstructures which are formed from different magnetic objects like scomions interacting with each other. And in some cases there are genuinely complicated 3-dimensional magnetic states which have never been imaged before. So in the first example, instead of a magnetic stripe, we used focused ion beam milling. This is a cooperation with Haifeng Du and Jiadong Zhang and colleagues to make a little cylindrical structure rather than a stripe. And if we image that cylindrical structure along its axis, then magnetically we see a scomion in the middle, and then the edge twist now forms a magnetic state around the edge which spins in the opposite direction. This little cylinder has a diameter of about 150 nanometers and a thickness of about 100 nanometers. The complexity, though, becomes apparent if we tilt this object about a horizontal axis. And I'm now going to show a tilt series of magnetic states. And these tilted magnetic states go down to about 60 degrees tilt about the horizontal axis. And whereas everything looks nice and symmetrical when you look along the cylinder along its axis, as it's tilted, you begin to see that magnetically the object is asymmetrical horizontally and it's asymmetrical vertically. In fact, it probably has a mixed block type and nail type character, which we would like to image in 3 dimensions, but that reconstruction hasn't been carried out yet. The second reason I mentioned is that we sometimes have magnetic singularities. And on the left, there are schematic diagrams which show a magnetic scomion tube or string going all the way through a thin TEM sample. And on the left, there is an equivalent object, which is called a chiral bobber, which ends in the middle of the sample at a singularity, which is referred to as a block point. These were predicted and then imaged using off-axis holography. On the right are phase images recorded in the electron microscope, and the red arrows point to chiral bobbers, which have the same size as magnetic scomions, but have weaker contrast, but they've never been imaged in 3 dimensions. And more recently, we had our colleagues predicted theoretically that if some magnetic scomions assemble together in a small close-packed arrangement, then at certain values of applied field, they don't form straight strings, which are seen on the right, but at lower values of applied field, they start to twist around each other to form what are termed braided rope-like superstructures. And these have also never been observed before experimentally, to our knowledge, to the best of our knowledge. However, if in the electron microscope, we again look at a very carefully machined lamella, in this case, a lamella which is 800 by 540 nanometers in size, imaged at low temperature in the presence of an applied field. And if initially 6 scomion strings are nucleated in this little iron germanium lamella, then they assemble to form a very curious structure which displays a magnetic phase profile, which is shown in the image on this slide. And a simulation of one of these superstructures, which is braided, where the magnetic scomion strings twist around each other, reproduces this curious structure. And so this is a direct experimental verification viewed in one direction of this scomion braid, but again, it's never been observed experimentally in 3 dimensions before. And finally, there are predictions of even more complicated magnetic states, which are referred to as magnetic Hopfeons. And here we'd like to image them not only in 3 dimensions, but we would like to image them interacting with each other magnetically in time and in space, as well as in 3 dimensions in the presence of external optical or electrical stimuli. And so 3-dimensional imaging of magnetic states is complicated, not only because, in contrast to normal tomography, 2 tilt axes of holograms are required, but in many cases, for example, with scomions, you need to be able to apply an external field and cool the sample to low temperature. So here I'd like to show some recent work which has been carried out by the group of Axel Lubk with Sebastian Schneider, Bernd Rellinghaus together with us. And here he overcame some of these problems by a very clever method, which involves making a ring 300 micron with a 300 micron hole in it of a hard magnet samarium cobalt, which is magnetized perpendicular to the plane, and then inserting a needle-shaped sample of iron germanium into this ring. This ring can then be cooled. The stray field of the ring exerts an external magnetic field on the needle-shaped sample, and that needle-shaped sample can then be tilted about 2 directions in the presence of an applied field at low temperature. And in this way, the 3-dimensional distributions of the 2 in-plane components of the magnetic field can be reconstructed separately, and then the criterion from Maxwell's equations that the divergence of B has to equal 0 can be used to reconstruct the 3rd component of B from the first 2 3-dimensional reconstructions. And this works very nicely. Here is a 3-dimensional representation of the in-plane components of B reconstructed in 3 dimensions. And I'll play a little movie here, which shows slices of the 3-dimensional distribution of Bx and By inside the sample, imaged as a function of distance Z through the needle. And if you may just be able to see that the centers of some of the scomions change in position as from slice to slice. What is very interesting from the three-dimensional reconstruction is that there are local deviations from block-like character. Some of the Scomions collapse at the surfaces of the sample. Some of the Scomions are not parallel to the applied field direction. And there are some correlated modulations of the positions of the centers of the Scomions from one Scomion to the next. And all of this is impossible to see unless you can reconstruct the magnetic vector field in 3 dimensions. This is with nanometer spatial resolution. So this is a reconstruction of the magnetic field B. The reconstruction of the magnetization M is a whole different problem. It's an inverse problem. And I just wanted to mention briefly that this is a problem which we are tackling in a different way, not by reconstructing the components of B separately, but by using model-based iterative reconstruction of the magnetization, effectively by guessing the 3-dimensional distribution of magnetization, and altering that guess for the 3-dimensional magnetization until it matches an experimental set of magnetic phase images. That allows us to introduce regularization terms and confidence parameters, as well as the known location of the magnetic objects inside the field of view, and it allows us to introduce additional constraints and additional help from machine learning and to incorporate micromagnetic simulations inside the reconstruction algorithm, which would be difficult with a normal back projection based tomographic reconstruction approach. I won't show any results from this today, but it also works very nicely to reconstruct M in 3 dimensions. And now, very briefly, I'd like to touch on the second and third topics before concluding. The second topic is switching of nanoscale devices in situ in the TEM. So here, as an example, I'd like to show phase change memory cells, which are small structures, again, only a few hundred nanometers in size, in which an electrical current can be used to change the device from an amorphous state to a crystalline state. The amorphous state has a higher resistance, the crystalline state has a low resistance, and they are of interest for high performance computing with reduced power, which is faster than, say, flash memory, also for applications as some kind of artificial synapses in neuromorphic computing. And we would like to understand how the devices switch on a local scale from between amorphous and crystalline states. So in this case, on the right-hand side here is an annular dark field STEM image of an individual memory cell. This is only 300 nanometers long and about 80 nanometers wide. And the challenge here is that we have to pass nanosecond pulses through the device. And this means that the specimen holder has to have impedance matched electrical connections. We have to have dedicated MEMS chips where the device works outside the TEM and inside the TEM. And we have to have additional layers to stop the device evaporating because there is no large heat sink underneath the device inside the TEM. So on the one hand, we can take images of the device switching between amorphous and crystalline states subjected to nanosecond pulses. And here we have different images where you can just about see which region is amorphous and which region is crystalline. But it's much better, instead of forming images like this, to take diffraction patterns point by point. And here are examples of diffraction patterns taken as a focus probe is stepped across the field of view. If we take diffraction patterns point by point, then first of all, we can make a direct map of which regions are crystalline and which regions are amorphous. In these images, everything which is green is a silicon nitride support, everything which is light blue is crystalline material, everything which is dark blue, which is amorphous. So in frame A, the bridge, the device is mainly crystalline, it's a low resistance state. In frame B, there is an amorphous region, a dark blue region in the middle of the bridge, and this is a high resistance state, and so on. But because we're recording diffraction patterns point by point, we can group together diffraction patterns which look similar, and we can look at the evolution of individual crystallites during switching and color code every region which is crystalline, which corresponds to the same crystallographic orientation. And in this case, as the device is switched between low resistance and high resistance states, you can see which crystalline regions remain in the same orientation as they are switched from cycle to cycle, and in which cases the switching is not reproducible. One of the reasons I show this is because in the future, we would like to see compositional redistribution from one cycle to the next, but separately, we would like to use phase contrast techniques like off-axis holography to look at the electric field distribution between the crystalline and amorphous regions when a voltage is applied to the device in situ in the TEM. And the final topic I wanted to touch on is electron beam sensitive materials. And here, I wanted to come back to a point which may not have been apparent from the beginning, which is that in phase contrast techniques applied at atomic spatial resolution, whether it's in high resolution TEM or whether it's in structural biology, we ideally want to image the phase of the electron wave, the phase shift, rather than changes in amplitude contrast. And a technique which allows us to record the phase directly is what we would ideally like to use, rather than, for example, defocusing and then inferring the phase shift indirectly. And I mentioned at the beginning that there are a number of different phase contrast techniques that we can use. If we look at electron beam sensitive materials, we are also interested in dose efficiency of these techniques. And here I'd like to illustrate the imaging of a very beam sensitive mineral sample. This is Beryl, beryllium, aluminium, silicon oxide. This is work carried out in cooperation with Bodil Holst from Bergen in Norway. And this particular structure has channels inside it. And not only channels, but it has individual atomic columns that comprise beryllium atoms, which are very weakly scattering, and the entire structure is very beam sensitive. The fact that the beryllium atomic columns are very weakly scattering is responsible for the fact that in high-end granular dark field images, the beryllium atomic columns are invisible, whereas in a phase contrast image, in this case, simulated for the technique of integrated differential phase contrast, the beryllium atoms suddenly are visible. These are simulations with no noise included, and the beryllium atoms are halfway between the black channels. Experimentally, this works very well. It's a very dose efficient technique. And on the top, I show 3 different images recorded using integrated differential phase contrast at biological electron doses of only a few tens of electrons per square angstrom. And if you look at the image on the top right, halfway between the black channels columns, the columns of beryllium atoms are visible. But not only that, if you take a Fourier transform of each of these images, then in the bottom row, the yellow circle marks 1 angstrom information transfer. And this means that at biological doses, we are getting close to 1 angstrom spatial resolution while being able to image very weakly scattering atomic columns with a phase-sensitive technique. In this case, integrated differential phase contrast, but it could be off-axis holography or it could be tychography instead. And this is, now I wanted to come back to the final topic here, which is future scientific directions here in our institute. Well, based on what the work with the examples which I showed, some of the directions in which we'd like to go in the future include moving towards imaging individual spins and charges on the atomic scale and in 3 dimensions in the presence of external stimuli, also with improved temporal resolution. These are all technical challenges. Some of them are more difficult than others, but there are other directions in which which would help us. And here I would say we would like to bring electron microscopes into the modern age. We would like to automate experimental workflows. We would like to do real-time data analysis. We would like adaptive optics, more dose-efficient imaging modes, better detectors, more correlative experiments. We'd like to have more modular microscopes and better specimen stages. And in Jülich, we are trying to go in some of these directions. In the short term, we are taking our microscope, which is used for imaging magnetic fields, and we're equipping it with an advanced magnetizing stage, helium flow cryostats, pulsed lasers, a beam blanker, a specimen holder that allows gigahertz pulses of current and fields to be applied, improved detectors, and automation software for both materials science and biology. But where I wanted to come back to, which I started from at the beginning about the large infrastructure perspective, is that we've succeeded in establishing ourselves on the national roadmap for large infrastructure in Germany. In other words, on the same funding stream that supports telescopes, satellites, and synchrotrons. And within this funding stream, we are ordering 5 unique electron microscopes. The funding agency, the ministry, said we can only order microscopes that are not currently being sold commercially and which will remain unique for 10 to 15 years. The first instrument is due for delivery in 2023 or 2024. That's a combined TEM and atom probe. This is a UHV instrument which is operated at helium temperature with 2 correlative techniques and 2 data streams coming at the same time inside a single instrument, perhaps even simultaneously. And then we have 4 further unique instruments which will be delivered in 2023 and 2025. And these instruments are currently under discussion with commercial manufacturers. So this is where I wanted to stop. And I wanted to, first of all, acknowledge all the people who've been involved in the examples which I showed. Some of these are people in our institute and some are external collaborators. And here I apologize if I've missed anyone out. And finally, I'd like to acknowledge the different funding agencies from the European Union, from the German Scientific Research Council, and we also have some funding from the US, from DARPA. And as a final slide, I just wanted to show a few examples of magnetic images of many different samples, which I didn't have a chance to show. So I will stop there, and many thanks for listening to that scattered range of examples.
Well, Rafal, thank you very much for the very exciting talk. Thank you. Yeah. A few questions due to the time limitation. Let me start from the first one. We're looking at the German's roadmaps on the infrastructure. I mean, that sounds very, very exciting. So one question is this 5 microscope and the planning. How much investment is that to get this 5 microscope are we talking about?
So in US dollars for the 5 instruments and including a building, it is somewhere between 100 and 150 million US dollars.
Okay.
And it's not so straightforward to raise the funding. It's an 8-year process to get to it.
Yeah. It's good to know also for the US side as well, what type of scale we need to talk about here. So that's good. Now let me ask you one question from Juan Carlos Idrobo from the audience. The question is, for the imaging of Scomions in 3D, what's the spatial resolution in 3 different directions? Is the difference in the in-plane direction versus the along the beam directions?
It depends on which approach you're using to do the reconstruction. If you are using conventional back projection based techniques, then you suffer from some missing wedge artifacts. And then the spatial resolution is necessarily better in some directions than others. In the plane, it is a few nanometers. Out of the plane, it depends on the tilt angle, but it could be worse than a few nanometers, maybe even a few tens of nanometers, depending on the tilt angles. If you're using model-based approaches for reconstructing the 3-dimensional magnetization or magnetic field, then you have additional constraints. And then the spatial resolution can be much more similar in the 3 directions. But again, it depends on the range of tilt angles you're using. So it's not a straightforward question. It's somewhere between a few nanometers and a few tens of nanometers, depending on the range of tilt angles.
Yeah. So Rafal, for this pillar, you are doing a Scomion study. If you have the size dependence, getting smaller and smaller, how do you expect the evolvement of these Scomion patterns? Would you go into it small and now you only have 1 in the center all the way going down instead of having 6 surrounding the center one?
So in the example which I showed, which was a single cylinder, that is a single Scomion with an edge twist around it. The size of the Scomion depends on the applied field, and it also depends on the material that you look at. In some materials, the Scomions can go down to a few nanometers in size, but then it becomes much more challenging to characterize them magnetically because the magnetic signal becomes very weak. So you can manipulate the sizes of the Scomions with applied field, and you can change their size. For the material which I described, each of the Scomions is about 70 nanometers in size and slightly smaller in the presence of an applied field. So when you make a sample which is smaller than about 100 nanometers, it becomes very difficult to nucleate a single Scomion inside it.
Yeah. Well, thank you, Rafal. I know our time is limited. Maybe one question for both you and Ada. If Ada's still online, I think this is from the audience. I like these questions. Which of the many options of future work are you most excited about? Maybe just pick one you are most excited about for both Rafal and also Ada, this question. Ada, you're on mute.
What do you mean by most excited?
I think this audience, maybe for Rafal, it's more like technique, right? What technique capability is most exciting to you? Ada, for you, it can be the scientific question. The
question is, which technique is most exciting?
That's probably more for Rafal. That's my guess. For Ada, it's probably the which scientific question is most exciting to you.
Which scientific question?
Yeah.
Everything that I don't know is interesting. I don't like the word most, because what is most today may be not most tomorrow, because I find another problem.
Yeah.
So most is a temporary description.
Yes.
Exciting is everything that I want to understand and I still don't know.
Very, very good, Ada. So Rafal, for you.
I would say exactly the same thing. I wouldn't focus mostly on techniques, because our technique development activities respond to new incoming physics and materials problems that people bring to us. So we only develop techniques to learn something new about the materials we study. So I would say it depends on what our collaborators bring us. And I would say with magnetic fields, there has never been a sample that we looked at where we saw what we were expecting to see.
Very cool. I want to get back to Bob and Wa and see whether you have comments, questions, Bob.
Thank you, Yi. Well, just fabulous talks, very stimulating. I have so many questions also. I'm particularly interested with Rafal about how resolution is measured in these circumstances, for instance. But what we should do is end fairly soon the recorded part of the session, and we'll stay online for people who'd like to answer questions. There are many questions on the Q&A, which we can transmit to you, and I hope you'll be able to respond to them separately. Or if anyone wants to stay online and ask you questions personally, we'll do that. But I think 2 fabulous stimulating talks, which I appreciate the time and effort for joining us today, and all the audience for participating as well. So I think that Wa has other circumstances that we might adjourn the recorded part of the session now. We'll look forward to seeing you on the first Monday of May with 2 more very exciting talks lined up. So thank you for the recording, and we'll stay online for more questions now. Thank you.